Good morning, everyone. I, Dr. Sakshi Bhardwaj, on behalf of Elton Technologies, welcome you all in this subsequent session of Women's Day special webinar series with today's topic Cheminformatics and Bioinformatics in the life and in drug development. Chemoinformatics or cheminformatics refer to use of physical chemistry, chemical data, and chemical information with computer resources, whereas bioinformatics is an interdisciplinary field that develops methods and software tools when it comes in terms of the biological data, right? So basically, there is, if I'm telling, there is no such difference in between the cheminformatics and bioinformatics, right? But these two fields are very important in terms of course, in the drug development as well. In today's masterclass, our guest of the day will be explaining how cheminformatics and bioinformatics plays the significant role in our daily life and in the drug development. It's giving me immense pleasure to introduce our today's guest of the speaker of the day, Dr. Sudipa Mondal. She is the founder and proprietor of Neurit Scientific and Computational Services. She is a PhD in chemical sciences with 5 years of postdoctoral experience in the field of drug discovery. Dr. Mandal has been graduated from Jadavpur University, Kolkata, West Bengal. Dr. Mandal is recipient of DAD by Nationality Supervised Doctoral Fellowship, CSIR Senior Research Associate Fellowship, National Postdoctoral Fellowship funded by Science and Engineering Research Board, National Eligibility Test conducted by Joint CSIR UGC, Rula International Award for Innovation in Chemistry in 2020. Dr. Mandal is a good combination of scientific organic chemistry and computational chemistry. She has her expertise, including computer aided drug designing, computational structure biology, bioinformatics, cheminformatics, and machine learning. She has expertise in handling different scientific softwares. She has earned 28 research papers and 509 citation to her credential. Dr. Mandal has industrial work experience as well. Now, without any further ado, let's start the session. We will be taking question and answer at the end of the session. So over to you, ma'am. Ma'am, you are on mute actually.
Yeah.
Yes.
Good morning, everyone. Thank you, all team, for this opportunity. Um Today, I will talk about chemoinformatics and bioinformatics in our daily life and its importance in drug development. So, in day-to-day life, we use lots of chemicals. Our life actually full of chemicals. To be true, whatever we are eating also a kind of chemicals only, though it is made by nature. Whatever we are eating, those are also chemicals, and many chemicals we use. Those are actually part of our uh, uh, hygiene and like hand wash, face wash, floor cleaner. Many chemicals we use actually every day, but very few of us have knowledge what are actually those things and how do they work. So today I will just give a brief discussion on those things, whatever the daily usage chemicals, and few, yeah, uh, and I will give just a brief description and how chemoinformatics is important in our life, and we should have knowledge about all those things to have a good life. Next, yeah, okay. I will mostly discuss on chemoinformatics and essential chemicals in our daily life. Then I will talk about bioinformatics and its role in our daily life, and then chemoinformatics and bioinformatics in the drug discovery. So let's start with the chemoinformatics. So chemoinformatics is an emerging science that concerns the mixing of chemical information resources to transform data into information and information into knowledge. Chemoinformatics is a tool that emerges from the combination of computer resources and chemical data and is used in the management, visualization, and systematic analysis of chemical information. It is actually a relatively new field of information technology that focuses on the collection, storage, analysis, and manipulation of chemical data. The chemical data of interest typically includes information on small molecule, formulas, structure, properties, spectra, and activities, biological and industrial. So, chemoinformatics is nothing but information about chemicals. So many databases are there that includes all kind data, includes information of almost all kind of chemicals we use. It could be a drug, that is, it could be antibiotic, it could be an antifungal drug, it could be something that we use for our personal hygiene, like that. Let's go for the next. Okay. All food items are nothing but some chemicals. Few are in very complex form, and we cook. Actually, nature prepare this in a complex form. We cook food in easily digestible form. Cooking is the process of producing safe and edible food by preparing and combining ingredients, and in most cases applying it. We cook by applying heat. Cooking is a means of processing food without which many foods would be unfit for human consumption. So basically, nature has produced very complex chemicals. By cooking, we actually we make it we make in a we get it in a digestible form. Again, what we do is, again, cooking is required for hygiene also. I mean, that is, many microbes may be there in the food. So when we cook by with heating, so the microbes are basically, they goes. Okay. Very common chemicals which are actually food items, like common salt, water, sugar, honey, curd, cheese. All these are basically nothing but some chemicals. Rice, dal, vegetables, edible oils are basically some naturally available chemicals. Few synthetic chemicals are there which are also part of our daily life, like toothpaste, hand wash, soap, face wash, face cream, detergent, different types of medicine. So I will just give brief information about those. Let's go for rice. Okay. Um, in rice, the main carbohydrate is starch. It is composed of amylose and amylopectin. The rice grain constitute 12% of water, 75 to 85% of starch, and only 7% of protein. So here is the structure of amylose. Every day we consume rice. So here just information that what actually rice contains. Rice contains mostly contains amylose and amylopectin. So amylose is a straight chain polymer, and amylopectin is a branch carbohydrate polymer. Okay, so these are the structure. So if anyone wants to go more detail in it, can go to the database and fetch more and more information. We know that different types of rices are there, like govinda bhog rice, badshah bhog rice. Different types of rices are there, and in different type of rices, this that amylose composition, I mean that percentage of amylose and percentage of amylopectin, it actually varies. and in some rices that water percentage is more, water percentage is less. So if someone wants to get more and more information, can go to the database and fetch more and more data and use that data. Human and other animals eat plant foods, and human and other animals that eat plant foods has an enzyme named amylase that helps in breaking down amylopectin to glucose. So whenever we are eating, our body already has a enzyme named amylase that actually break down this amylopectin or amylose into glucose, and we get the energy. So chemoinformatics will say you what is the structure of amylose, okay, what is the structure of amylopectin, and the bioinformatics will tell you what is that enzyme amylase is, what is its structure, how it works, like that. Now, wheat, that is also a very popular source of carbohydrate. Wheat flour mostly contains carbohydrate, but it contains a protein named gluten. I think many people knows about the gluten. The hard flour, wheat flour are of two types. One is hard flour, another is soft flour. So it basically depends on the percentage of gluten. If the percentage of gluten is more, then it is the hard flour. If it is less, then it is the soft flour. Wheat gluten is composed of mainly two types of protein, the glutenin and the gliadins. The gluten peptides are responsible for triggering gluten related disorders. But few disorders are also reported, that is, those are called the gluten related disorder. That is, few people has intolerance with the gluten. Okay, those people are basically then whenever and actually for them the gluten free wheat is suggested for them. Okay, gluten related. So if someone is having this kind of disorder or someone having this kind of intolerance, can go through this data and can get more information. Okay, gluten related disorder is the umbrella term for all diseases triggered by gluten, which include celiac disease, non-celiac gluten sensitivity, wheat allergy, gluten ataxia, and dermatitis herpetiformis. Okay. Similarly, we can get more information if someone wants to get more information about the dal. There are different kinds of dal. So dal contains what if it is how many percent, how much percentage it contains protein, carbohydrate. Anyone can get that kind of data. So all these data are available in the database. Here is a structure. The structure actually represent a part of the gluten protein, that is from the 43 to 49. It is showing, see, the high percentage of proline is there. So it is basically combination of both chemoinformatics and bioinformatics. Okay, from bioinformatics, we have to know the sequence of the gluten protein, then its structure, then we will analyze. So during this analysis, chemoinformatics is required. We can see that high percentage of proline is there. So this proline may be the reason of for those people who has the gluten intolerance. Okay, now every day we take fruits. So I think we also should know what are the antioxidants are. We all have heard that we should eat fruits. It has antioxidants, and all. So here I have chosen apple first. Apple contains a variety of phytochemicals, including quercetin, catechin, fluoridazine, and chlorogenic acid, all of which are strong antioxidants. Apple contains malic acid also. We all know that apple contains malic acid, but apple contains these antioxidants also. And these are the structure of the antioxidants. This is quercetin, this is the structure, this is catechin, and this is fluoridazine. Okay, structures are quite similar, but differences is also there. See here, C double bond O is there. This C double bond O is missing here. Here, see, a double bond is there in the quercetin and the catechin. This double bond is not there, but this is a but this carbon is again it is actually a chiral carbon. So this kind of important information we can get from chemoinformatics. Here, interesting data is here. So here actually antioxidant activity of various fruit extracts. So in a paper, I found that the antioxidant is maximum in the cranberry, followed by the apple, then red grape, then strawberry, peach, lemon, pear, banana, orange, grapefruit, and the pineapple. Okay, so from this data, you can get which fruit has maximum amount of antioxidants. Excuse me, please. Grapes are a rich source of bioactive molecules, including phenolic acids, flavonoids, anthocyanins, steel beans, and lipid. So, in my last slide, I just gave picture of the antioxidant present in the apple. Similarly, antioxidants are present in grapes. Those structure is also available. Banana also have some bioactive compounds. So, if one is interested to know what are the bioactive compounds are there in the banana, can get it. Okay, banana contain phenolics, carotenoids, biogenic amines, and phytosterols. Okay, so I briefly discussed about whatever the food items we are taking and what are the chemicals present, chemicals or the antioxidant present in the food items. Now, in our day-to-day life, few chemicals are part of our life. We cannot live without them. And so I will just briefly say what are those things. Okay, toothpaste. So toothpaste is made of calcium carbonate, sorbitol, sodium lauryl sulfate, sodium silicate, sodium monofluorophosphate, sodium bicarbonate, carrageenan, etc. So why these things are required? Okay, so just let me explain very briefly because these are not such thing that I can explain it in very 5 minutes, but it is just introduction. Okay, carrageenan is used for as a thickening agent. Okay, and now sodium lauryl sulfate is a surfactant. It cleans away the dirt inside the mouth. Bicarbonate is one of the salivary components that potentially modifies the formation of caries. It increases the pH in saliva and in this way creates a hostile environment for growth of acidic bacteria. So sodium lauryl sulfonate and bicarbonate is used so that acidic bacteria cannot generate inside the mouth. Okay, but fluoride is most important. Fluoride is considered to be the most effective caries inhibiting agent, and almost all toothpaste today contain fluorine in one form or the other. So all the toothpaste actually contains the fluoride. Without fluoride, no toothpaste is effective. The most common form of sodium fluoride, actually mostly sodium fluoride is used, otherwise monofluorophosphate. Okay, so if you check that Colgate mostly contains monofluorophosphate, other toothpaste may contain sodium fluoride also. So see, since sodium fluoride is very effective, so sometimes sodium chloride also may be effective. So you might have noticed few ads that do your toothpaste contain salt. So yeah, it can contain sodium chloride. So if we add sodium chloride, it could be more effective. Okay, so whatever the day-to-day things we are using, I think it's good to have little bit of knowledge of that. Now, face wash, shampoo, and hand washes. Sodium dodecyl sulfate or sodium lauryl sulfate is the main ingredient in shampoo, face wash, and many hygiene products. So this is the structure of sodium lauryl sulfate. It has a hydrophilic head. See, this is the hydrophilic head, and it has a hydrophobic tail. Now, ACS forms that sodium lauryl form, and the question is how it works. So whenever we put sodium lauryl sulfate in the water and we rub our hands like that, so what happens? Whatever the hydrophobic impurities are there, excuse me, this, whatever the impurities are there, it actually form a micelle. So see this ball kind of things it actually forms, where the hydrophobic tail is inside and the hydrophilic head actually in the outside, and the hydrophobic impurities, I mean that dart actually goes here. So all the hydrophobic impurities get soluble in this way, and when we wash it after rubbing, so it goes with that, it goes with that one. So we get clean hands, clean face like that. Shampoo also works in the same way. SGS from micelle in water. It has a hydrophobic core. Oil or dart goes inside the micelle. Micelle have widespread use. So micelle activity is not only limited in this washing or the cleaning. It has a widespread use in drug delivery to carry a series of different molecules in it, including low molecular mass hydrophobic drugs, proteins, and genes. So this micelle activity is used in drug delivery also. Okay, now floor cleaner. Floor cleaner also part of our life and we use it. So uh, this is actually benzalkonium chloride. It is a cationic surfactant and it is the main ingredient of Lysol. So I'm not advertising for any brand, but I'm just giving an example because Lysol is one of the most popular brand for the floor cleaning as a floor cleaner, and I'm just want to say that it has the benzalkonium chloride, and it is a cationic surfactant, and it also works in the similar way um, as a micelle, and it removes the dart. Okay, and we know that a toilet cleaner is Harpic. Harpic actually mostly contains the hydrochloric acid, and the another type of Harpic is called the red Harpic. Red Harpic actually contains sodium hypochlorite, NaOCl, or it is called bleach also, along with sodium hydroxide. And very popular disinfectant or antiseptic liquid is Dettol. Dettol actually contains uh, chloroxylenol. Okay, so this is the structure. Okay, one benzene ring is there, one OH group is there, and one chlorine group is there, and two methyl group is there. So, uh, chloroxylenol is the main ingredient of the Dettol. Savlon is also very popular antiseptic liquid, and it is basically combination of two chemicals, that is cetrimide and chlorohexidine. So this is the structure of chlorohexidine, and this is the structure of cetrimide. Now come to the medicines. Medicines also are chemicals that are part of our daily life. Okay, let's have a look on the medicinal plants. We all know that neem is an important medicinal plant, and here I have just neem plant in has I mean it contains many chemicals, and one the most the percentage highest percentage is uh, aja azadirachtin. It is the main active substance. So this is the structure, and it is the structure of nimbolinin. It is actually a bigger structure. It is not easy to remember. It is just information that these are the uh, these are the chemicals that are inside the neem plant. Okay. So let me very briefly say about a few medicine that is part of our life. Antacids, antacids, pyrogesic, antibacterial drug and the antifungal drug. Okay, so this is pan 40. I think oh, most of us are very well known to pan 40 or pan D. So the main ingredient in the pan 40 is pentoprazole. So this is the structure of pentoprazole. Pentoprazole is a type of medicine called a proton pump inhibitor. So its mode of action, how it works, it actually works as a proton pump inhibitor. Pentoprazole covalently binds with proton and H plus or K plus ATP pump to inhibit gastric acid and basal acid secretion. The covalent binding prevents acid secretion for up to 24 hours or longer. So pentoprazole basically works through the proton pump, works as a proton pump inhibitor, and it uh, stops the secretion of gastric acid. So bioinformatics is, so chemoinformatics will say you about the structure of the pentoprazole, and bioinformatics will tell you about the how it works. Okay, how it inhibits. Again, now paracetamol. So this is the structure of paracetamol or acetaminophen. Mode of action, actually paracetamol has a widespread use, but its mode of action is not very clear. But it is believed that acetaminophen works upon the brain to block prostaglandin production. Okay, so few evidences are there that it may be working as a prostaglandin production. It blocks the prostaglandin production. So this is the structure of paracetamol. Next, sulfamethoxazole. It is a very old drug, and I am telling about nowadays. In very few cases, sulfamethoxazole is used, but I am telling about here because I started my journey in this drug discovery with the sulfamethoxazole. My whole PhD work is on sulfamethoxazole. Sulfonamide drugs were the first broadly effective antibacterial, and and it paved the way for the antibiotic revolution in medicine. So whatever the antibiotic revolution has occurred so far, actually it started with the sulfamethoxazole. In bacteria, sulfonamides act as competitive inhibitor of the enzyme dihydroxytrate synthase, an enzyme involved in folate synthesis. So dihydro dihydroxytrate synthase is a enzyme. So the sequence and the structure of the enzyme we will get in bioinformatics. If this enzyme is active in the bacteria, and the bacteria required required folate synthesis for its survival. So dihydroxytrate synthase actually binds with para-aminobenzoic acid. Okay, to synthesis for the synthesis of folate. But what sulfamethoxazole do is sulfamethoxazole works as a competitive inhibitor of the para-aminobenzoic acid, and it binds to dihydroxytrate synthase before the para-aminobenzoic acid. Thus, it stops the folate synthesis. So it is the structure of sulfamethoxazole. Now, ketoconazole. Ketoconazole is an antifungal medicine, and it is used for many kind of antifungal infection, including the dandruff. For the treatment of dandruff, actually ketoconazole is mostly used. So this is the structure of ketoconazole, ketoconazole, and again another one. Yeah. So ketoconazole has uh, used. It interferes with the fungal synthesis of ergosterol by inhibiting the enzyme cytochrome P450 14 alpha uh, demethylase. So this is the enzyme how ketoconazole works. Yeah, I have drawn two structure because there is a chiral center here. and their chiral center here also. So these are actually a mixture of enantiomers. So I drawn two structure. So few open resources are there to get information about any chemical, any kind of chemicals, and those are gene database, PubChem, ChemSpider, Drug.com, and many other open resources are also there. Now bioinformatics. So, bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data, in particular, when the data sets are large and complex. Bioinformatics combines biology, chemistry, physics, computer science, information engineering, mathematics, and statistics to analyze and interpret the biological data. So few open resources are there to for bioinformatics, NCBI database, Swiss Prot, DDBJ, and PDB database. Any information related to protein, DNA, RNA can be obtained by bioinformatics. Sequence of a protein, its secondary or 3D structure, conserved domain, multiple sequence alignment, drug protein interaction can be obtained by bioinformatics. Actually, whatever the resources I said you, that NCBI database, Swiss Prot database, protein data bank, it contains so much. It contains lots and lots and lots of data. So you will get almost. I mean, you will get almost all kind of protein sequence, secondary structure, tertiary structure, like that. So I'm just giving an example, like ubiquitin. Okay, so I think many of us has heard about the rheumatoid arthritis. This rheumatoid arthritis is due to the malfunctioning of ubiquitin producing system. So if somebody wants to know what is ubiquitin, so you can get the sequence. So see, here is the sequence of the small protein ubiquitin. Okay, CMQIFBKTA. So this is the amino acid sequence, and an amino acid is represented by one letter. So this is the total sequence, and it is the structure of human ubiquitin, the available in the protein data bank. I've just given the. I have already worked in the ubiquitin system, so I have just showing you the structure. Okay. So in previous slide, I showed that how to fetch the sequence, how to get the structure of any kind of protein. Okay. In the bioinformatics, you can also get information how a small molecule interact with a protein. Okay, so see, it's a structure of a dihydroxide synthesis. Yeah, the structure of dihydroxide synthesis, and inside this, one small molecule is there. It is actually sulfur methoxazole. So if we study this structure, I mean, if we visualize this structure very closely, we can see how actually this is interacting with this protein molecule. And to visualize this one, softwares are required. Okay, so many softwares are there to visualize this one. Uh, PyMall, Discovery Studio. These are very good for visualizing this kind of structure. Okay, now bioinformatics and chemoinformatics in drug development. So in my previous slides, I discussed how to fetch information about the chemicals, how to fetch information about the proteins, and now how to use this one for the drug development. So first of all, an extensive literature review is required to choose the target pathway. So which target pathway to which target protein or which pathway to select, we have to do a extensive literature search. So many open resources are there to do the literature review. Here we have chosen Fanconi anemia pathway. I have already worked with the Fanconi anemia pathway. I have papers on it. So after extensive literature review, we have chosen this pathway. Okay. Now let me tell about this. So Fanconi anemia pathway is a major DNA damage repairing pathway. So DNA may get damaged due to many reasons. Okay, due to it basically it could be damaged by some drugs or anything. So if some DNA get damaged, our body has a system that it will repair that DNA. Okay, so Fanconi anemia pathway is a very complicated pathway that repairs basically the damaged DNA. So this is the intercrossing damaged DNA. Here it is the structure, and the damaged DNA then undergoes that Fanconi anemia pathway. So Fanconi anemia pathway is a complicated, and 22 genes are involved in this pathway. Once the DNA goes through this pathway, after that it get repaired. So this repairing process is very complicated. So after doing many kind of study, so I have already said that 22 genes are involved in Fanconi anemia pathway. We have chosen only one protein. Actually, we have chosen many proteins, but here I will discuss about only one protein. Okay, so we have chosen FANC-L. So here I'm just showing you the cryo-electron microscopy structure of core Fanconi, the Fanconi anemia core complex. Okay, Fanconi anemia core complex. It is just to show that how complicated actually the original thing. This complex is available in the protein data bank, but analyzing this one is little difficult, and for this we need the software. So bioinformatics actually requires many kind of softwares to analyze this kind of complex, and we have to choose one or two protein because we cannot work with all the protein at a time. It's not possible. So it is a structure of core complex, a complicated structure. Okay, so after extensive literature search, we have chosen FANC-L. Here is the structure of FANC-L. So FANC-L crystal structure is reported, but the total protein is not reported. Actually, part is reported. One crystal structure reports the first part, C-terminal part. Another crystal structure reports the middle one. So what we did it, we need to model this one using all the crystal structures available. So here is the structure of Franklin, and this is the Ramachandran plot. Yeah. In the literature, we found that FANC-L form a complex with E2 conjugate. Okay. So here is the structure. This blue part is part of the FANC-L, and the purple one is part of the E2. So we found that I mean interaction is there between FANC-L and E2. So we thought that we will break that one. So we may be successful in our, whatever our aim. Okay. So I'm just showing you how to choose that binding domain. So this is the Frankel. Here we have chosen the binding domain. Basically, can you see this sphere? The bigger sphere is those part that where the E2 conjugate is there, and others are also in the cavity, so we didn't left that, we use that one also. Then we take the database, chemoinformatic database, small molecule database we take, and then we did the virtual screening, and we got that few compounds are showing good docking score. So it is the details docking picture. So whenever we get a good docking score, depending upon the docking score, we will choose few compounds. Okay, so here are the three compounds we have chosen depending on the docking score and its admit property. And based on this, we have to choose that, okay, this is the best structure. Maybe then we have to synthesis it, and we have to do the sale assay. After the sale assay result, we can report. Okay. or you can take the bit like that. Okay, so that's all. Thank you. Thank you for your kind attention, and thank you all, Tim, for the opportunity.
Thank you, ma'am. That was very informative session in terms of explaining the cheminformatics, how each and every day we are playing with the chemicals, like in terms of whether it is in vegetables, fruits, any kind of shampoos, hand washes. Yes, exactly. Those are having those kind of chemicals which we are using in our daily lives, right? And how these bioinformatic tools and technologies we are using for the drug development process as well. So let's start with the taking some questions. We have some questions, so can we have those questions?
Yeah.
Okay, the first one is, what is the greater than symbol in starting out that faster sequence?
Actually, FASTA is a general format, and it starts with the greater than sequence. I mean, those people who have started the FASTA sequence, they introduce it one. So it is
uh,
the rule that if it is a FASTA sequence, then it will start with the greater than sequence. If it is the greater than sign is not there, then no software will detect it as a FASTA sequence. You will get the error, and you cannot upload it.
Exactly.
This
That
is
is
very important.
basically the descriptive information we can say that
Yeah, yeah, yeah.
symbolize that
It is actually
descriptive.
like, yeah, it is actually given to the, I mean, those scientists have introduced who has actually introduced the FASTA sequence.
Okay. Next question. How Ramachandran plot could be helpful in analyzing protein structures?
Yeah, Ramachandran plot is actually very good. He actually, I mean, Dr. Ramachandran, he actually made this database that if that Shai belongs to this zone, if Phi belongs to this zone, then the protein structure is valid. If the Shai and Phi and doesn't fall in this particular zone, then it is, then that structure is not valid. Okay, so Ramachandran plot is very important.
Yes, actually, it is showing that torsion angles in between the amino
Yeah,
acids,
yeah, yeah,
how
yeah,
they are.
yeah, yeah.
Yes, and
Actually,
basically
torsion, yeah, yeah,
analyze.
toxic, yeah, yeah. It should fall in particular region. It cannot fall here and there. It cannot be scattered.
On the basis
So
of their
yeah,
stability, we can say that we
yeah,
are analyzing the protein structure while seeing these Ramachandran plots, basically. Uh, Next question. Okay. What could be the workflow to start a drug development process, basically?
yeah, yeah. First, you have to start the literature review is the first thing. That from where to start. Okay, then you have to go step by step.
So starting from the mining of your structures, then
Yeah,
with your
yeah,
proteins, and then the homology modeling techniques we are doing, pharmacophore mappings we are doing, until the interaction, docking, and the simulation. So
yeah,
all these comes into the drug development process, basically.
yeah, 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 yeah.
Uh, Next question, please. Do
Drug,
we have
drug repurposing is also there. For that,
yes,
you don't need to. Then, yeah, then using chemoinformatics you can do that. I mean, if it is drug repurposing.
yes, exactly. Okay. Why is it important to check for the ADMET properties of a drug?
Yeah, admit property is very important for a particular drug. So whatever we are designing, it should be absorbed. It should be distributed in the body. Then it should go properly in the metabolic pathway. Then it should be excreted also, and it should not be toxic. So all these databases are required. Suppose we are giving a drug which has a excellent absorption, distribution, but it is not excreted properly. It is staying in the body, then it should not be a good drug. Okay, and it should not be toxic. It should not be teratogenic. So admit property is very important. It is actually, yeah, after designing, those are very important. And chemoinformatics is important. Chemoinformatics is important in that part.
We have done with all the questions, ma'am, for the session. Yes, thank you all. It was very informative session given by ma'am actually to how she is. Means I would say that chemistry, it's the beauty of science basically. How each and every component is having those chemicals in, and you are dealing, each and every second with those chemicals by knowing or unknowing things, so which we are, and developing technologies like the bioinformatics and the cheminformatics we are using for our drug development process. So yes, we are at that advanced stages of these kind of technologies. Yes. So thank you so much, ma'am, for connecting with us for this webinar, and thank you all for joining us today for this session. So thank you, ma'am. Thank you.
Yeah, thank you. Thank you all, team. Yeah.